nanotechnology has various applications in several fields. Here we can synthesize several types of nanocomposites or nanocompounds or several other nanomaterials having potential application in biomedical fields, that means nanomedicine, in the field of photocatalysis for the development of some photocatalytic nanomaterial to degrade the organic pollutants from the wastewater to make the water pollutant free, and also the several types of novel nanocomposites and nanocompounds having very good supercapacitive activity, good thermoelectric activity, etc. But there are different types of process of synthesis of different nanomaterials. But the method of synthesis can be classified into two groups. One is chemical method of synthesis, as for example the Sol-Gel method, hydrothermal method, etc. And another is physical method of synthesis. That means the mechanical alloying method or ball milling approach. Chemical method means basically the bottom-up approach, whereas the physical method is top-down approach. In our lab, we widely use the physical method like mechanical alloying or ball milling process. Here we have two model of ball mill. One is P5 ball mill and another is P6 types of ball mill. And here we now showing the entire process of this ball milling method. At first, we take the precursor powder in required molar ratio. That means, as for example, if we want to synthesize the TiO2 Ag nanocomposite with 9:1 molar ratio, then we should calculate the required weight of TiO2 and required weight of Ag for the synthesis of these types of nanocomposite, and then we take this amount of TiO2 and Ag by using a balance, and then we should use this type of mortar pestle to mix it, and by mixing it for about 30 minutes we can make a very good mixture of these two precursor powder. And when the mixture gets ready, then we can transfer it into the chrome steel vial or chrome steel pot of that ball mill. This is basically the chrome steel pot, and this contains around 30 chrome steel balls. And after the transferring of this precursor powder within this chrome steel vial, then we place this vial inside the ball mill. Okay. And this is our ball mill. This is basically our P6 ball mill, P6 model of ball mill. And this basically planetary ball mill. This is called planetary ball mill because it has planetary like motion. The chrome steel vial will rotate around its axis in one direction, and all the media will rotate in opposite direction. That's why it has planetary like motion. That's why it is called planetary ball mill. So at first we place the vial with the precursor powder in the ball mill, and then we should tight it very carefully to avoid any types of accident. Okay. And then we should also fix the RPM of the rotation and also the time of the rotation, time interval of the rotation. And when we fix all these parameters, then we should close the ball mill, and then we should click on the start button to start the ball milling process. Okay. After the ball milling of several hours, that means at around 3 to 4 hours, we can prepare these types of nanocomposites, and we should collect it and then send it for the several structural and microstructural study. That means we can characterize it by several microstructural study like X-ray diffraction study, SEM study, TEM analysis, etc. So this is basically one pot synthesis method. Without using any expensive chemical, we can synthesize any types of nanocomposite or nanocompound in a very short interval of time. If we use various types of chemical method, then we have to use several types of lengthy steps, and we have to use several types of expensive chemicals. But in this process, we only use the precursor powder, and within very short time interval, within 3 to 4 hours, we can synthesize this type of nanocomposites or nanocompounds without using any other expensive chemicals. That's why it is a very simple, facile and cost-effective method for the synthesis of several nanocomposites. Okay. Now we are discussing about other chemical method. Okay, like other Sol-Gel method, hydrothermal method, etc.
One of the sample preparation method is sol-gel process, and in this video we are going through this process step by step. So at first we are trying to prepare BiFeOBi nanocomposite. So for the preparation the required samples are bismuth nitrate, and this is the bismuth nitrate. Next is the ferric nitrate. Next one is the KBr, and next is ethylene glycol, and this is the water. At first, all the beakers are washed repeatedly with acetone and then dried, and then 60 ml water is poured into a beaker, and in another beaker there are 60 ml of ethylene glycol. Next, we have to weight these samples one by one. First, we take a butter paper and placed on the weight machine. Then we can tear, and next 0.727 bismuth nitrate is required. Now this bismuth nitrate is added to the ethylene glycol solution. Next we have to measure the ferric nitrate among about 0.636 gram. Now this 0.636 about this ferric nitrate is again poured into the ethylene glycol solution. Next we have to measure the KBr. This KBr is added to the 60 ml water, and then stir properly to mix all the solution, and then this KBr solution is added dropwise to this ethylene glycol solution. Now this KBr solution is dropwise added to this ethylene glycol solution, which is already placed in a magnetic stirrer and in a water bath at 80 degrees Celsius. We can see that after a few times the color of the solution will change, and next we are continuously stirring for 5 hours. Now the color of the solution, you can see that the color of the solution has changed. Now to change the pH of the solution, few ml of ammonia is added, and you can see that the color of the solution is again changes. And now we continuously stirring it for 5 hours at 80 degrees Celsius. One of the most promising method to get morphology of the sample is hydrothermal method. Now we are showing you the entire hydrothermal process. Here we try to synthesize nickel cobaltite nanowire. For this the required chemicals are nickel nitrate, cobalt nitrate, urea and ammonia. First we have to measure these samples in appropriate manner. We take 1.5 gram cobalt nitrate. First we take 1.5 gram cobalt nitrate and it is added to the water solution. Now we take 0.75 gram nickel nitrate. Next this nickel nitrate sample is poured into that solution. Next the mixture is stirring for another 1 hour. After 1 hour, 1.2 gram urea is weighted and added to the solution. And then it is kept stirring for another 2 hours. Next, few drops of ammonia is added to the solution to change the pH of the solution, as well as the color of the solution has also changed. After 2 hours, the solution beaker tightly sealed with a plastic so that no vapor can enter into it. This is the autoclave. Then the autoclave is switched on and wait until the pressure reaches to 1, and then it is kept at 120 degrees Celsius for another 6 hours. Then the beaker solution is taken out from the autoclave and then repeatedly washed with alcohol and ethanol, and then it is filtered. Then the sample is dried at 60 degrees Celsius and then annealed for 2 hours at 350 degrees Celsius. We achieve the final product, that is the nickel cobaltite nanowire.
After the successful formation of the nanocomposite by several routes, that means several physical and chemical routes, now the structural and microstructural characterization of the sample is needed. For the microstructural characterization of the sample, the XRT pattern should be taken. And here now we are showing the entire process of the X-ray diffraction. And this is our XRD lab. Here basically for the X-ray diffraction, we basically used the X-ray pattern are basically recorded with the X-ray powder diffractometer. Here the model is Bruker D8 Advance, and it is operated at 40 milliampere and 40 kilovolt with nickel filtered CUK alpha radiation. This is basically the sample holder or mounter, and at first it should be cleaned very properly for avoiding any type of contamination, and then the sample should be placed at the center of this holder. This is the sample. In this way, in this manner, the sample should be placed at the center of the sample holder, and then by placing it very carefully, the upper surface of the sample should be smooth, and then it is taken inside the sample, inserted into the onto the sample holder of the X-ray diffractometer, and then it should be adjusted very properly. In this manner it should be adjusted very properly, and then the diffractometer start to record the data, and on the monitor we can get the X-ray diffraction pattern. This type of X-ray diffraction pattern, and here this is a plot of between 2 theta versus intensity, and from this plot we can identify the several crystalline planes that are present into the sample, and from this we can confirm the several constituent phases which are present in the sample. This is basically a sample XRD pattern, and this is the XRD pattern of CuZNO nanocomposite. In this CuZNO nanocomposite, we can see that several types of planes are present of copper and zinc, and also a CuZn alloy. In this way, in this manner, we can identify several constituent phases which are present in the nanocomposite. Again, for the structural and microstructural characterization of the sample, again a very important procedure is scanning electron microscopic technique. It is a type of electron microscope that produces the images of the sample by scanning the surface of the sample by focusing the beam of electrons. Here the surface will be scanned with the beam of the electrons. These are basically the FESM images, and by these FESM images we can calculate, we can measure the particle size, and these are the particle size histogram. From the FESM images we can see that from these FESM images we can see that there is a very good morphology, surface morphology. So for several times this type of surface morphology can have a great application in several fields like supercapacitor application, etc. Okay, and these are basically the particle size histogram which can be obtained by measuring the particle size from these same images, and by this way we can understand the morphology of the sample, and also we can measure the average particle size by this analysis of the scanning electron microscopic technique or measurement. Okay, and now we will discuss about the transmission electron microscopic technique. For the detailed structural microstructural characterizations of the sample, at first we have to measure the average particle size of the sample and also the nature of the sample. That means if the sample is crystalline or it is amorphous, right? Thus, for this microstructural characterization, that means to measure the average particle size of the sample and to understand the nature of the sample, the best technique that we can use is the transmission electron microscopic study, right? This transmission electron microscopic study has several number of steps. At first, we have to prepare the sample. At first, we have to prepare a sample solution. That means we have to prepare a solution of the sample in the ethanol, a well dispersed solution of the sample. And then we have to take a carbon coated copper grid, and on that grid we have to take a small drop of the sample, and the grid then should be dried overnight, and then the grid is sent for the TEM study, right? Here the whole process of this TEM study is now showing in this video. So at first we have to prepare the sample for the TEM study. So to prepare the sample solution, at first we have to take some amount of ethanol in the test tube, and then a very small amount of sample, pinch of sample, very small amount of sample in the ethanol, right? Then for making a well dispersed solution of the sample in our lab, we basically use a bath sonicator. And in this bath sonicator, the sample, this solution, this mixture of the ethanol and the sample is sonicated for, has to be sonicated for a long time. That means at least for 5 hours for getting a well dispersed solution. And then after the formation of this well dispersed solution, here we take a 300 mesh carbon coated copper grid, and then we take a drop of that well dispersed solution on that grid and dried for overnight, and then this dried grid was sent for the, or was used for the TEM study, right? Now for the TEM study, here basically a beam of electron is used for this TEM study, and this in this TEM study we can use two modes. One is the transmission mode and also another is the diffraction mode. Okay. To measure basically the particle size, we can use the transmission mode. And for understanding the nature of the sample, that means if the sample is crystalline or it is amorphous, we can use the diffraction mode. Okay. Here we are showing the whole procedure of this TEM study. This is basically the sample holder, and on the sample holder we have to take the dried grid very carefully. We should do it in very careful manner, because to avoid any rupture of that grid. And then we should place the grid, the dried grid basically, the dried grid on the sample holder very carefully, and then this sample holder, and then we have to insert the sample holder. Okay, then we can get the TEM image for the transmission of the electron beam and the diffraction of the electron beam through the sample, and these are the TEM image, and this is basically the SAED pattern. So to discuss about this whole process, this is basically in this way the electron beam basically transmit through the sample, and then we can get the TEM images. Now The wavelength of the high energy electron is a few thousandths of a nanometer, and the spacing between the atoms in a solid is about 100 times larger. Thus the electrons are diffracted and the atoms act as a diffraction grating. Thus a fraction of the electrons will be scattered to particular angles determined by the crystal structure of the sample, right? While other pass through the sample without reflection. The resulting TEM images will be a series of spots constituting the selected area diffraction pattern. This is basically the selected area diffraction pattern. Here we can also get a series of spots or some rings, right? Each spot corresponds to a satisfied diffraction condition of the sample's crystal structure. If the sample is tilted, different diffraction conditions will be satisfied and different diffraction spots will appear. And for this ring pattern, each ring designates a particular crystalline plane. As for example, this is for the copper 200 plane, and we can identify the particular plane by measuring the d spacing, that means the interplanar spacing of the plane. And if we get these types of rings or spots, then we can say that the sample is crystalline. By this way, we can understand the nature of the sample. That means the sample is crystalline or it is amorphous in nature. All this can be done in diffraction mode. But if we use the transmission mode, that means the transmission of the electron beam through the sample, then we can get the magnified images of the particle in this way. Okay. And from these images, TEM images, we can easily measure the particle size and can plot and can and also plot the diameter of the particle and also the number of the particle, and we can have a particle size histogram. From this particle size histogram, we can easily measure the average size of the particle. So by this way, in this manner, by transmission electron microscopic study, we can do a detailed characterization of the sample and also can find the average particle size of the sample and also can understand the nature of the sample, that means if the sample is crystalline or not, and we can also identify the several crystalline plane of the sample, and also if we prepare some types of nanocomposite, then we also can identify the several constituent phases that are present in the sample by this by identifying the crystalline planes. Okay. Now I have already discussed about the synthesis process of the nanocomposite, that means the NC sample, and the drug conjugated nanocomposite sample, right? That means the NCDC sample. Okay. And I have already discussed about the various types of its structural and microstructural characterizations of these samples. Now the main aim is to apply them in several biological fields, like in the several biomedicinal fields. So we have applied these types of drug conjugated nanocomposite as antibacterial agent and also as antifungal agent, anti-diabetic agent, etc. in various biological fields. So at first I am discussing about the application of this type of drug conjugated nanocomposite as antibacterial agent. So here we have basically used two types of bacteria like E. coli gram-negative bacteria and Staphylococcus aureus like gram-positive bacteria. And we have used the agar cup diffusion method. Here we have prepared an agar cup and then apply both the nanocomposite, drug conjugated nanocomposite and also the pure drug. This black shaded region basically gives the region in which the growth of the bacteria was totally inhibited due to the application of the paste compounds, right? So by measuring the diameter of this zone of inhibition, we can confirm that the nanocompounds can be used as antibacterial agent. So here we can see that the nanocomposite, pure nanocomposite, that means without any conjugation of the drug, the diameter of the inhibition zone is significant. That means only the pure nanocomposite can also be used as a very good antibacterial agent without using any external drugs, right? We have also tested the drug conjugated nanocomposite sample in same method, that means agar cup diffusion method, and by this method, by measuring the diameter of the zone of inhibition, here we can see that for NCDC sample the diameter of the zone of inhibition is greater than the pure drug. But I have told that in the NCDC sample only 5 weight percent of the drug is present. That means only the 5 weight percentage of drug can have the same activity as the pure drug when it is conjugated with the nanocomposite sample. This is the basic novelty of our work, and by this way we can successfully synthesize a very good antibacterial agent with a minimum side effects to the normal human cell, right? We again confirm the antibacterial activity of the nanocomposite and the drug conjugated nanocomposite, and also we have compared the antibacterial activity of the drug conjugated nanocomposite with the pure antibiotic by MIC study. That means the minimum inhibitory concentration study, which is the concentration in which the optical density of the bacteria is nearly equal to 0. And here from this study we can say that the MIC for the NCDC sample is about 20 microgram per ml, and for the drug it is 80 microgram per ml. That means there are 40 folds more significant activity for NCDC sample than the pure antibiotic. This again proves that the antibacterial activity of the NCDC sample has been enhanced in a significant manner, right? Now, we also apply other types of nanocomposite as synthesized, mechanosynthesized CuAg-ZNO nanocomposite and drug conjugated nanocomposite. Basically, the drug like antifungal drug fluconazole conjugated nanocomposite as a very good antifungal agent against various types of fungi like Candida species. And again by the same procedure, by measuring the diameter of the zone of inhibition, we can say that the NCDC has very similar activity to the pure drug fluconazole. But in NCDC sample, only 5 weight percentage of the fluconazole was present. That means the activity of the drug is significantly enhanced when it is conjugated with the NC sample, right? It again confirmed by the minimum inhibitory concentration study as I have discussed already, and here again we have confirmed that the 5 weight percent drug can produce the same effect as the 100% drug does when it is conjugated with the nanocomposite. That means the drug conjugated nanocomposite can be used as a very good antifungal agent, and by this way the dose of the drug can be minimized, and also the side effects and the cost of the drug can be minimized when it is conjugated with the very cost effective method with some non-toxic nanocomposite, right? Again, we have also synthesized basically various types of non-toxic nanocomposite and again conjugated it with some anti-diabetic drug like metformin. And we have applied it in two types of mice. One is normal mice and also one is hyperglycemic mice. That means the mice having a hyperglycemic tendency. And after the treatment, we can say that the NPDCX means the drug conjugated nanocomposite sample when it is applied in the same concentration as the same concentration of the pure metformin sample. NPDCX has the similar activity as the same antidiabetic activity as the pure drug metformin. But in metformin, pure metformin, 100% metformin was present, but in NPDC example, only 5 weight percentage of the metformin was present. That means the 5 weight percentage of metformin can show the similar effect as the pure 100 weight percent metformin when it is conjugated with these types of non-toxic nanocomposite, right? Now, we have also tested these types of various compounds against the normal human cell to confirm the non-toxicity of the material, because the cytotoxicity is a very important factor, because here we want to synthesize some types of nanomedicine which are totally non-toxic, having a minimum side effects to the normal human cell. And from the cytotoxicity test, we can see that the percent cytotoxicity is almost less than 15%. Thus, it can be considered that the samples is totally safe and non-toxic to the live cell. So, by this way, by this very cost-effective way, very simple method, one-pot synthesis method, basically very facile method, we can synthesize this type of nanocomposite, totally non-toxic nanocomposite to the normal human cell, and also conjugate the drug with the nanocomposite for the synthesis of some drug conjugated nanocomposite, which has a very good antibacterial agent, and which can also be used as very good antifungal agent. And again, these types of nanocomposite and drug conjugated nanocomposite can also be used as very good anti-diabetic agent, and by this way we can minimize the side effects of the drug in a very simple method, right? Okay, thank you.